Pigments. Pigments is an HL topic for the biochemistry option, and it's a little bit of a varied group. There's going to be really three different categories of pigments that you have to look at. Pigments are molecules that cause things to be colored inside of plants or animals. We've learned a little bit about color previously in the course. We've learned that things appear colored when they absorb certain parts of the visible spectrum. And so generally what we are seeing is we're going to see the complementary color to what is being absorbed, the complementary or the reflected color. Most pigments that we're going to look at, or actually all of these pigments that we're going to look at, are strongly colored because they have a structure that is what is called highly conjugated. So that's going to be an extremely important term to be able to accurately use. They are highly conjugated systems. So this means that there are a series of alternating double and single bonds, generally between carbon atoms, but not necessarily between carbon atoms. Could also be with nitrogen or oxygen. You're also going to have them connected to benzene rings, and that's going to allow electrons to delocalize across a very large number of atoms. Remember that when you have one double bond, that's because you have a set of parallel p orbitals. But if you have two double bonds near one another, so this is one double bond with their p orbitals, this is another double bond with these p orbitals, the p orbitals can actually delocalize across that single bond that's in between. And so in fact, it ends up acting like one sort of continuous pi bond across all three of those bonds. So we've talked about that before when we looked at resonance structures. This is electron delocalization. And so we've mainly looked at it in terms of like a benzene ring. We've looked at it for maybe a couple of bonds at a time. Well, the pigments that we're going to look at have systems where the pi bonds are conjugated over many, many, many atoms. So it's a little bit different and unique. Now, if you think about it, the more delocalized the electrons are, so that if there's more and more bonds that they can move between, they are less and less strongly held to any one particular position within a molecule. So this means it takes less energy to sort of excite them, because if they're held down really tightly to something, really tightly in one place, or localized in one place, it requires more energy to cause changes in them. But if they aren't,
I need my glasses to read.
So if they're held down in one position, then it takes a lot of energy to excite them, to cause them, or only high energies will make, they will only absorb high enough amounts of energy, often which are in the ultraviolet spectrum. But if you can delocalize them and they take less energy to excite, then the type of energy they absorb will actually be in the visible spectrum. So when these electrons have visible light shown on them, some of the wavelengths they can absorb. That causes them to be excited and move to slightly higher energy states. And so as they absorb those particular visible wavelengths, they end up reflecting the complementary colors. So it produces very strong visible colors in these pigment molecules. The part of the pigment molecule that actually does the absorbing of the visible light is called the chromophore. Before we get into the actual pigments, you are expected to be able to discuss two analysis methods for pigments. The good news is they're pretty much exactly the same as what you already went over for proteins and the analysis of amino acids. There's both paper chromatography and something called thin layer chromatography. They are almost identical, except paper chromatography uses a piece of chromatography paper as its stationary phase, while thin layer chromatography, TLC, uses a thin absorbent layer of silica or alumina. So these are like silicon and oxygen, or aluminum and oxygen compounds that are sort of made into a paste and spread across a piece of glass or plastic. They work under the same principles. You spot something at the bottom at what we'd call the origin. So you could mark a line in pencil, and then you're going to spot your sample here. So you'd spot like something, a biological sample that's colored. Now, often things that are colored contain multiple pigments, and so you're going to spot them onto this either this sort of paste, this plate as we call it in TLC, or the piece of paper. And then you're going to add a solvent, and the solvent will not reach that origin. But that's like the worst liquid line of all time, but you get the idea. So you put it in a solvent. Solvent's going to slowly soak up the plate, and it's going to carry the different parts of the pigments, the different pigment molecules, to different distances. And so you might get some green, a green spot up here, and you might get a yellow spot, a little bit further down. And then you might get a different green sort of here. And so depending on how polar they are, how soluble they are in that particular solvent, those things move up the sheet, the plate, to different degrees. You can mark off that solvent front, and then you can calculate RF values. The same way we did before. You'd measure the distance of the spot and the distance of the solvent front, and you divide. The advantages of thin layer chromatography over paper chromatography is that it's faster. It works pretty well with small samples of pigment, and it produces more reproducible results than paper chromatography. Otherwise, the steps are the same as if we had done paper chromatography for amino acids. The first category that we're going to look at for pigments are the carotenoids. So if you've ever heard of beta carotene, that is a carotenoid. There are a couple of different carotenoids that are going to show up in the data booklet, both alpha and beta, and they're generally associated with yellow and orange colors mainly in plants, but they can produce colors ranging from yellow to red. They're referred to as an accessory pigment in photosynthesis. Usually these things are also in green plants, but you don't have as much of them as you do of the chlorophyll, and so plants still appear green, but they do have these like yellow and orange pigments in there as well that absorb different parts of the visible spectrum, and so they help transfer energy to the chlorophyll and make the chlorophyll more efficient. They have these really long hydrocarbon chains, and that's where the conjugation is taking place. So if you look at the alternating double bonds here, and you imagine the pi bonds, well, we've got a pi bond here, and then there's a pi bond here, and then there's a pi bond here, and I think you get the idea. Pretty much every other bond is a carbon-to-carbon double bond, and that's just going to create a conjugated system throughout, all the way from here and then through this entire molecule, all the way across that hydrocarbon chain. You'd have electron delocalization. Okay, so a tremendous amount of delocalization means that those electrons are easily excited, and so it's going to absorb visible light. These things, if you take a look, it should be fairly easy to spot that they are completely fat soluble because they are made only of carbon and hydrogen. So they're going to be very nonpolar, have lots of London forces, and so they're going to be fat soluble, not water soluble. They can't form hydrogen bonds with water. These things these are precursors for vitamin A. If you look at the structure of vitamin A, it pretty much looks like one of these things chopped in half. And so they're very important for proper vision, getting enough carotenoids, things like carrots that contain them. I mentioned before that they are in green leaves, and one of the reasons why in the fall, when there's less and less light and it starts to get colder and plants are going towards their winter mode, the chlorophyll sort of degrades, and that's when you start to see these carotenoids in the plants. They are still there, absorbing different parts of the spectrum, and that's when the plant's leaves look yellow and orange. These things are relatively reactive. We've talked about before, especially we talked about fats, that carbon-to-carbon double bonds are fairly reactive. So these alpha and beta carotene can react with oxygen from the atmosphere, and that makes them effective as antioxidants. It means they themselves can go bad, but it means that they can protect other important particles from going bad. So it's aided. These oxidation reactions are aided by light, and so if you expose these things to less air, and if you keep them in the dark, it tends to make things containing the carotenes last longer. And if you eat the fruits fresher, it means that you'll have better nutrient content in your food. And so sometimes the oxidation reactions, as they did with the oxidative rancidity with the lipids, they can give off volatile chemicals which cause bad smells, but they can also cause changes in color and a loss of vitamin A activity. Okay, the second category that we'll look in this video are the anthocyanins. Anthocyanins are generally associated with red, pink, and blue colors in foods. If you've ever seen like red cabbage or cranberries, things like that, then you were looking at some anthocyanins. They're formed by the reactions between sugar and proteins. And so as fruits ripen and they get more and more sugar concentrated inside of them, it tends to lead to more anthocyanins in the fruit, which is part of the reason why fruits get more and more colored as they ripen most of the time. They are aromatic compounds. Aromatic is a term in the organic chemistry unit that refers to things that contain phenyl groups or benzene rings. And so the conjugation occurs across both the phenyl groups, but also in these other rings that have alternating carbon-to-carbon double bonds. And they call it a C6, C3, C6 structure. It's a little bit hard to spot at first, but you have this ring, which contains 6 carbons. So that's the C6. And then you have these 3 carbons right here. So that's a C, right? That's a C, and that's a C. So that's 3 carbons. So there's the 3. And then the aromatic benzene ring contains the other 6. So those things the anthocyanins tend to have in common. And you'll notice that there's alternating double and single bonds throughout, some of which are connected to oxygens, some of which are part of this benzene ring. But that's a highly conjugated system, which gives it the color. These are from the data booklet, and these are 2 forms of the same anthocyanin in different pHs, which we'll talk about in a bit. One use of the anthocyanins is that they can be used as pH indicators. Many of you may have in middle school taken red cabbage and blended it up and used it as a natural indicator for pH. What happens is that the hydroxyl group over here in the anthocyanin can act as a base, or sorry, can act as an acid, can donate protons if it's in a more basic environment. And so as that H plus gets lost, there are a number of ways that the electrons will sort of shift around. And so many of the double bonds end up changing locations in what we call the chromophore, in the part of the structure that gives it the color. So if you change all the locations of the double bonds, it changes the exact wavelengths of light that are absorbed, and so the color of the anthocyanin changes. The generalization for the acid-base colors, as you've kind of already learned, is red is acidic, so you should know that one. They tend to be a little bit more purple when neutral, which is kind of how you often see them in the vegetables themselves. And then they actually turn sort of a yellow-green when basic, or sometimes we say a blue when basic. So they're used as pH indicators. So here is the acidic version of the anthocyanin. It's got the proton right here. Now, what I've drawn is if that proton is lost, so here's now the donated proton over here, what that would leave behind is an O minus. Well, the O minus can, one of these lone pairs, which is contributing to the minus charge, can move and go down to the bond between the carbon and the oxygen instead, creating this carbon to oxygen double bond. Well, if that double bond goes there, that would give this carbon atom 5 bonds. So then this double bond has to move. And you can see how this ripple effect works. So this double bond moves here. So then this double bond has to move and go onto this oxygen, which gives that oxygen a neutral charge. And so those changes end up producing this molecule. So it's just that one donation of a proton, and then the electron pairs moving 1, 2, and 3. And so sort of shifting that charge around, so it makes the overall charge of the molecule neutral. And now you have this different structure. Different conjugation means a different color. So this one changes from red at the high pHs, where this is protonated. If you make it at higher pHs, more basic, the proton will get donated off, and the electrons will kick down and create the blue form. One other factor that affects the coloring of anthocyanins is that certain metal ions can bind with them, like iron 3 ions and aluminum ions, that you might find if you were canning fruit. You would have cans made out of aluminum, or possibly that contained a little bit of iron. And so if metal ions are being a little bit dissolved off of that metal interior, they can form complexes with the anthocyanins that give an even deeper color. So sometimes canned fruits look a little bit darker and richer than the fresh fruits they come from, and it's part of the interaction of those metal ions with the anthocyanins. There's a third category of pigments called porphyrins, and we're going to treat them in a separate video.